Welcome back, guys. Now let's begin with the muscle physiology. In this muscle physiology, first I will be explaining in detail about the skeletal muscle. Later we'll be discussing about cardiac muscle as well as the smooth muscle. Okay, three important muscles that you have to know in the muscle physiology: skeletal muscle, cardiac muscle, and the smooth muscle. Now, first let's begin with the skeletal muscle, which is most important for your exam. Okay. First of all, why a muscle is called as a skeletal muscle? Skeletal muscle, because it is attaching with the skeleton. So the muscle which is attached to your skeleton or your bones is called as a skeletal muscle. So it helps in the movement of the bones. Okay. Now skeletal muscles, they are voluntary in nature. Voluntary in nature means they are under your control. Okay. If you wish, you can contract them. Now in a skeletal muscle, what are the different parts present? Look at here, guys. Here I am showing a skeletal muscle. Let's say this is a muscle. Now in this muscle, okay, in this muscle, what is present? In the muscle, there are these fascicles present. Okay, there are these fascicles present. Now I am taking one such fascicle outside. Okay, one such fascicle outside. Okay, now here is a muscle, and some basic points are there. Like these are very basic points. Like muscle is covered by epimysium. Okay, this entire muscle. Okay, this entire muscle is covered by what? This entire muscle is covered by an fascia called as epimysium. Epimysium. Okay, and here there is perimysium. Around the fascicle, what is there? Around the fascicle, there is perimysium. Okay, there is perimysium around the fascicle. Okay, this all this area which I am highlighting, this is called as what? Perimysium. Now look at the fascicle. Inside the fascicle, what is there? In the fascicle, within the fascicle, there are these muscle fibers present. These muscle fibers. What are all these, guys? These are all muscle fibers. Okay, and every muscle fiber, okay, every muscle fiber is surrounded by endomysium. See, around the every muscle fiber, there is a covering called as endomysium. You can see this endomysium. This is all endomysium. So here there is endomysium. So there is epimysium, perimysium, endomysium. Okay, here there is epi, peri, endomysium. Epimysium is surrounding the entire muscle, epimysium. Fascicles are surrounded by perimysium. And these muscle fibers are surrounded by what? These muscle fibers, individually they are surrounded by endomysium. Now this muscle fiber which I am taking out, I am taking one muscle fiber outside. One muscle fiber. The one muscle fiber is nothing but a myocyte, one muscle cell which contains actin, myosin, which will contract. So within this muscle cell or muscle fiber, what do you have? You are having this myofibrils. Myofibrils. What these myofibrils are made up of? Sir, one myofibril is made up of actin, myosin, which are nothing but myofilaments. So what is the order, guys? Muscle. Within the muscle, fascicle. Within the fascicle, muscle fiber. Within the muscle fiber, myofibril. Myofibril made up of actin, myosin, called as microfilaments or myofilaments. Okay, so this is the order. Here it's already given. Within the skeletal muscle, what do you find? Muscle. In the muscle, fascicles. Inside the fascicles, there are muscle fibers or myocytes. Within the muscle fiber, there are myofibrils. Myofibrils are made up of what? Actin and myosin. Actin, myosin. That's it. This actin, myosin together called as what? Myofilaments. Okay, they are individually myofilaments. Right. Now, if I am starting with the muscle cell membrane, okay, if I have to start with the muscle cell membrane, look here. This is the muscle cell membrane. Let's imagine this is the sarcolemma. This is the sarcolemma. This is the muscle cell membrane. This is outside the cell. This is the inside, extracellular side and intracellular side. Now, within the muscle cell membrane, okay, you have to know some important proteins are there. What are the important proteins? Look at the first protein. See, this is a protein which is there from outside to inside. It is extending from outside to inside. So this protein is called as integral protein, sir. What is the name of this integral protein called as? Dystroglycan. So there is something called as a dystroglycan, there is something called as a sarcoglycan, and there is something called as sarcospan. So tell me, what are the three important integral proteins which are present in the muscle cell membrane? Dystroglycan, sarcoglycan, sarcospan. So that's what I am showing here. The three important proteins in the sarcolemma are dystroglycan, sarcoglycan, sarcospan. All are examples of what? IP stands for integral proteins. Next up, sir, there are two proteins which I am highlighting in a different color. What are they? Syntrophin, dystrophin. Where the syntrophin, dystrophin are present, you can very clearly see here there is a syntrophin and here there is dystrophin. So the syntrophin and dystrophin, they are present only towards the inside the cell membrane. They are only attached towards one side of the cell membrane, only inside. So they are called as the peripheral proteins. So PP, peripheral proteins. Okay, so this is what I want you to know, the five important proteins: dystroglycan, sarcoglycan, sarcospan. Dystrophin and syntrophin. Dystrophin deficiency will lead to disease called as Duchenne's muscular dystrophy. Already we have discussed Duchenne's muscular dystrophy. Weak muscles will be seen. Okay, complete absence of dystrophin, something like that. Now, within this diagram, you can very clearly see. So this protein called as a dystrophin. Okay, here is a dystrophin. No, so this dystrophin, it is attached with what? Intracellularly, see, on one side it is attaching with the dystroglycan. Let's say on one side it is attaching with the dystroglycan or the cell membrane. On the other side, look, this dystrophin, it is attaching with what? Actin. Cytoskeleton actin is there. No, I have told you in every cell there is actin present. So dystrophin intracellularly it is attaching with what? Actin. So that's why dystrophin attaches with actin intracellularly. Important. Next, after this, let me explain you about the sarcotubular system. Okay, sarcotubular system. Okay, already here the image is there, but let me draw the image for you so that you will have a clarity. You will get a clarity. So everyone, try to draw this diagram with me. Guys, here the structure which I am showing here in black color. So what is this? This is your muscle cell membrane. Here is a muscle cell membrane. In certain places, this muscle cell membrane, it is forming this invagination. Like this. And again, let's say here one more such invagination is there. Means the cell membrane is actually going into the cytoplasm. So this invagination which I am showing here, this is called as what? T-tubule. So this invagination, what do we call this invagination as? T-tubule. So let me write here, this is T-tubule, transfer, T for transverse, T-tubule. Here is a T-tubule, transverse tubule. Now, let me show you what is L-tubule, guys. L-tubule. The structure which I am showing you here in the red color. This structure. This is called as what? L-tubule. So there is something called as a T-tubule and there is something called as a L-tubule. T for transverse, L for longitudinal tubule. Now, you will have it out. Sir, we know what is T-tubule. T-tubule is nothing but the cell membrane, invagination of the cell membrane. Then what exactly is this L-tubule, sir? L-tubule is nothing but sarcoendoplasmic reticulum. This is nothing but sarcoendoplasmic reticulum. Sarcoendoplasmic reticulum. Okay? L-tubule. Now, within the sarcoendoplasmic reticulum, what is there, sir? What is there? See, there is a protein. Okay, let's say there is a protein. To this protein, who is attached? Let me draw it. See, here I am showing a protein. So, to this protein, who is attached? So, to this protein, these ions are attached, which I am showing in the red color. This is a protein which is binding with some ions. Which ions are these? These are calcium. So, there is a protein which is binding with the calcium. So, the blue color protein which I am showing here, this is a calcium binding protein called as a calsequestrin. So, right, what is the name of this blue color protein, guys? Calsequestrin. Calsequestrin is binding with what? Calcium. It is binding with which ions? Calcium. So, some calcium ions are also free. So, now tell me, what is the function of sarcoendoplasmic reticulum? The sarcoendoplasmic reticulum, it's a storehouse of which ions? Calcium ions. It's a storehouse of calcium ions. Lots and lots of calcium is present over here. So, calcium is not freely available in the cytoplasm. Calcium is not freely available for the muscle, guys. Otherwise, the muscle will be kept on contracted state. Calcium is available only whenever there is a necessity. So, calcium, all the calcium is tightly packed over here. So, this is all calcium. So, now tell me what is the calcium binding protein called as? The name of the calcium binding protein is called as a calsequestrin. Also, there is one more protein called as a calreticulin that I will discuss later. For now, calsequestrin binds with the calcium. Done. So, T-tubule completed, L-tubule completed. Now, let me explain you about the three important receptors here. In this sarcotubular system, three important receptors I will explain you. See, the first receptor that I am showing here on the T-tubule. See, on the T-tubule there is a receptor which I am showing in the green color. What is the name of this receptor? The name of this receptor is called as DHPR, dihydropyridine receptor, or dihydropyridine calcium channel. DHPR, it's a calcium channel, sir. DHPR, dihydropyridine receptor. So now tell me, DHPR is present on which tubule? T-tubule. And on the L-tubule, there is one more. Okay, there is one more channel, which I am showing here in the blue color. So this channel is called as the RYR channel. RYR, Ryanodine receptor. Ryanodine receptor, DHPR and RYR. Now, one more channel I am going to talk about. Here I am drawing in the brown color. It is also present on the L-tubule. What is the name of this one? The name of this channel is called as, actually it's a pump, sir. This is Sarcoendoplasmic reticulum, calcium ATPase. Sarcoendoplasmic reticulum, calcium ATPase. Okay, there are 3 pumps: DHPR, DHPR, RYR, and Cerca. Okay, 3 things completed. Now, let's see what is happening in the cytoplasm. Guys, in the cytoplasm of the muscle, okay, in the cytoplasm, there is a zigzag protein present, okay, which is in the shape of Z. So, this zigzag protein, okay, it's actually a protein. It is forming this zigzag line. Okay, so this line is called as a Z line. It is called as what? Z line. It's actually made up of a protein. Later I will tell you what is the name of this protein. The name of the protein is called as alpha actin. It's a protein. Okay, so here also one more Z line is there. Here also one more Z line I am showing. So 2 Z lines are there. Now the area between these 2 Z lines, is called as sarcomere. The area between these 2 Z lines is called as what? Sarcomere. So in between the 2 Z lines there is a sarcomere. Okay. Next, sir, to these Z lines what is attached? So to these Z lines, okay, even before that, let me show you one more. See, in the middle of the sarcomere, there is one more protein present, like this, which is forming a line. Actually, it's a protein. So this line is called as the M line. This line is called as what? M line. So in the middle of the sarcomere, there is M line. So students will start to have a doubt, sir, what exactly is the sarcomere mean? Sarcomere is considered as a functional unit of a muscle. Functional unit of a skeletal muscle, sarcomere. So there are Z lines and there is a middle में M line. In the middle there is M line and periphery there are Z lines. Now look carefully. To this Z lines, who is attached, guys? There is a protein that is attached here, like this. To these Z lines. What is the name of this protein called as? The name of this protein called as actin or thin filament. So these filaments which I am showing here, they are all actin filaments. Okay, these are all actin filaments. So actin filaments are attaching with what? Z lines. And to this M line, who is attached? So to this M line, there is a thick filament attached. The name of this thick filament is called as, look here, look carefully, this thick filament is called as myosin. So here also you can find the actin. These are also actin. So this is a different sarcomere. It will come into the different sarcomere. Okay, now you understand? So there are sarcomeres present. Now one sarcomere is considered as a functional unit. It's a contractile unit. Okay. Now tell me, what are the boundaries of the sarcomere? Periphery. In the periphery there are Z lines. In the middle there is a M line. Who is attaching with the M line? Myosin. Okay, this is the filament which I am highlighting here in the red color. This is myosin filament. Myosin is a thick filament, and this red color filament which I am highlighting here right now, this is actin. Now on the actin, what are they? Sir, on the actin there are active sites present. Actin contains active sites. Okay, actin, what do they contain? They contain these active sites. Okay, actin contains active sites. Then what about the myosin, guys? Myosin, they contains myosin heads. Myosin contains what? Myosin heads. So on the myosin, here I am showing you, these are the myosin heads, like this. So myosin heads are there. Now always these myosin heads, what they want to do, the myosin head want to go and bind with active sites. They will go and bind with the active sites, and they will try to pull the actin filaments. Now tell me, whenever they are trying to pull these actin filaments, the actin filaments will come towards the center. They will try to come towards the M line. So what will happen to the sarcomere size? the sarcomere size will decrease. The sarcomere size will decrease. So if the sarcomere size decreases means muscle will contract. Muscle will contract. So now you understand. But the question is, why during a normal relaxed state, why this myosin heads, why they cannot go and bind with the active sites? Okay, whenever the muscle is contracting, see, whenever a muscle is contracting, this myosin heads, they will go and bind with the active sites and they will pull the actin filaments towards the center, so that the two Z lines will come close together, so that I can say the sarcomere size is decreasing, muscle is contracting. But my question is, why during resting phase, the myosin heads is not going and binding with the active sites? It's because the active sites are actually closed. The active sites, there is a filament which is blocking the active sites like this. There is a filament which is blocking the active sites. What is the name of this violet color filament which I am showing here? This filament name is called as a tropomyosin. Tropomyosin. So what is the function of the tropomyosin, my dear students? Tropomyosin blocks the active sites, doesn't allow the myosin heads to go and bind with the active sites. Next up, anything else that you should know? See, there is one more protein which is attached with the tropomyosin. What is the name of this protein? Actually, it is having 3 subunits like this. What is the name of this protein called as? This is called as a troponin. The wheel-like protein is called as a troponin. So there is something called as a troponin as well as a tropomyosin. Okay. So now what and all we have discussed? There is something called as a T-tubule, L-tubule. There are 3 important channels called as DHPR channel, RYR channel and Sarca. Sarca is a pump. It's a Sarcoendoplasmic reticulum calcium ATPase. One is present on the T-tubule, other 2 are present on the L-tubule. Now, what exactly is the sarcomere? The area between the 2 Z lines is called as a sarcomere. Sarcomere contains what? 2 contractile proteins called as the actin, myosin. These are the contractile proteins, and 2 regulatory proteins are there. What are the 2 regulatory proteins of muscle contraction? One is the tropomyosin and other is the troponin. What is the function of a tropomyosin? Tropomyosin covers the active sites. And what is the function of the troponin? I will tell you, it will actually bind with the calcium. So now, let me tell you more parts of the sarcomere. Okay, more parts of the sarcomere. Guys, listen carefully. See, the area between this Z line from here, the area between the Z line till here, guys, till here. Okay, what is this area called as? Now let me use the dotted line. So this area. So in this area, within this area, only which filaments are present? Actin. Only actin filaments are present. So this area is called as what? This area is called as I band or isotrophic band. Okay, I band. This area is called as what? I band. I means isotrophic, means only one type of filament is present. Okay. Next step. Tell me, what is this area called as? So this area, okay, actually it is from here till here, but I am only showing on one side. Okay. Let's say from here till here. What is this area called as? See, within this area, actin, myosin. Both filaments are present, both actin is present as well as myosin is also present. So this area is called as the A band, means anisotrophy, not same, anisotrophic band. Okay, so this is called as the A band. Okay, there is I band, Z band, I band, A band. Now, tell me, what is this area now? From here, okay, from here only till here. What is the central area? See, in this central area, only myosin. Only myosin is present. How I used to remember, myosin is something, is not a thin filament, definitely it's a thick filament. Thick means heavy. So, heavy for H. So, in this area, see this area that I am keeping the dots. So, this area of the sarcomere, where only myosin is present, this is called as H zone. In the center, there is H zone. Now, in the center of the H zone, which line is present? In the center of the H zone, there is M line. So, why this is important for your exam? Okay, why this is important for your exam? They will ask you, arrange the order from the periphery to center in a sarcomere. Arrange from periphery to center in a sarcomere. So, always remember Z-I-A-H-M. Z-I-A-H-M. So, you can remember like Zia. Z-I-A. Zia is the name of a girl. Okay, Z-I-A. Zia. Z line, I band, A band, and H-M. Okay, you marry her, she will become your home minister. Okay, she will take care of you home. So, Zia, Z-I-A-H-M. Remember it like that, from periphery to center. Okay, done. Now, how exactly muscle contraction is going to occur? Now let me tell you how exactly, these are the components of the sarcomere, done. How exactly muscle contraction is going to occur? Let me tell you clearly. Let's say here is the neuron. Okay, here is the alpha motor neuron that I am showing you. This is the lower motor neuron or alpha motor neuron. Already I have told you, so this alpha motor neurons, what do they release? See, whenever they are getting depolarized, what is they going to release? They are going to release the acetylcholine in the neuromuscular junction. Now this acetylcholine, it will come and act on the muscle. It will come and act on the acetylcholine receptor or NM receptor. Now tell me, this acetylcholine, where it is coming and acting, guys? The acetylcholine is coming and acting on a receptor called as NM receptor or acetylcholine receptor, you can say. Nicotinic, NM stands for nicotinic receptor on the muscle. Okay, now? So now this acetylcholine is coming and acting on the receptor. Now whenever the acetylcholine is available, this receptor, which is an ion channel, so this acetylcholine receptor is actually an ion channel. What it will do? It will allow the sodium ions. It will allow the sodium ions to go into the cell. So sodium is coming into the cell. So lots and lots of sodium comes into the cell. So because of this sodium, there is a depolarization. Means the voltage is becoming positive. Because of the sodium, see, sodium is moving in the cell. So because of this, there is depolarization of the muscle cell membrane. The muscle cell membrane is getting depolarized. The voltage is changing because of this positive ions, sodium ions. Now you tell me, whenever this depolarization or the action potential, you can say, whenever this action potential is spreading along the muscle cell membrane, it will also go over the T-tubule. Now on the T-tubule, who will recognize this voltage change? Who is acting like a voltage sensor? Now this depolarization, it will come and open, or it will come and activate the DHPR channel. So who is acting as a voltage sensor for this depolarization? DHPR channel. Now the funny thing is, this DHPR channel, it is actually having a hand, sir. The DHPR channel, what it is having a hand, let's say. It is having a hand. Now whenever this DHPR channel is activated, now with this hand, mechanically, see, the word is important, mechanically, the DHPR channel, it will come and it will open. It will come and open the RVR channel. So mechanically, it will come and open the RVR channel. So now tell me, who is open for the first time? Now for the first time, this RVR channel opened. Now tell me, when the RVR channel is opened. Okay, first of all, this event is called as what? Electro, means because of the electrical activity. Electro. Mechanically, the DHPR channel opens the RVR channel. So this is called as electromechanical coupling. So what do we call it, guys? We call it as electromechanical coupling. Okay, EMC, electromechanical coupling. So now you tell me, electromechanical coupling, what exactly is happening during the electromechanical coupling? The DHPR channel will open the RVR channel. Once the RVR channels are opened, who will come out of the cell, my dear students? You guys are experts. Now tell me, who will try to come out of the cell? Sir, all this calcium, which is there in the sarcoendoplasmic reticulum. So calcium will come out. Calcium will come out. So for the first time, who is available? For the first time, calcium is available for the muscle contraction. Calcium. Now what this calcium will do, guys? So electromechanical coupling helps in the release of calcium. Again, we will discuss this later. But general out figure I am giving you. Okay, idea. So now this calcium, what it will do? Now the calcium, it will come and bind with. The calcium is coming and bind with a protein called as a troponin. So calcium comes and binds with what? Very, very important. Calcium binds with troponin. Calcium binds with the troponin. Recently asked. Calcium binds with what? Troponin. Now, you try to imagine, whenever the calcium is available, this is like a wheel. No, try to understand, this wheel will rotate. Whenever the calcium is available, what this troponin will do? Let's imagine it's going to rotate, sir. Now, whenever it is rotating, what it will do? Sir, it is attached with the tropomyosin, no? So, the tropomyosin is taken back. Imagine there is a wheel to which tropomyosin is attached. Whenever the wheel is rotating, the tropomyosin is taken back. So, for the first time, this tropomyosin, it will be moving away. So, the tropomyosin is moving away, exposing what, sir? Now, exposing the active sites. Now, the active sites are exposed. Now, this myosin heads will go and bind with the active sites, and with the help of ATP, they will pull the actin filaments, causing the muscle contraction. Now, you understand? So, again, one by one. So, what exactly is happening during the muscle contraction? Skeletal muscle contraction. Whenever the lower motor neurons or the alpha motor neurons, whenever they release the acetylcholine acts on NM receptor or acetylcholine receptor. Sodium will come into the cell, causing the muscle to undergo depolarization. The depolarization is detected by DHPR channels. DHPR channels mechanically open RVR channels. RVR channels will release calcium. Calcium comes and binds with Troponin. Troponin, what it will do? Troponin will remove the tropomyosin away, exposing the active sites. When the active sites are exposed, the myosin heads will go and bind with the active sites. With the help of ATP, they will pull the actin. They will pull the actin towards the center, leading to muscle contraction. Okay. So finally, can I say something like this? Electromechanical coupling. What and all I have explained you here is a sarcotubular system. Means sarco means muscle. So in the muscle there are T-tubule, L-tubule. Okay, sarcotubular system I have explained you. Along with that, I have explained you about the components of the sarcomere. Okay, components of the sarcomere. Also, I have explained you about the electromechanical coupling. Now, guys, you tell me, once the muscle contraction is completed, let's say muscle contraction completed, sir. Now, do you want this calcium to be available in the cytoplasm? No, we don't want muscle contraction completed, so calcium should have to go back. So, how this calcium is going to go back? See, now this calcium, once it is done, once its function is done, now this calcium, look, now what happened to this calcium? The calcium will again go back. The calcium is again sequestered back with the help of Sarca pump. So what is Sarca is doing? Sarca, is it helping in muscle contraction or relaxation? Muscle relaxation. It is sequestering. It is sending the calcium back into sarcoendoplasmic reticulum. So the three channels are completed: DHPR channel, RYR channel, and Sarca. So Sarca is a relaxing protein. It is helping in the skeletal muscle relaxation. Okay. Now, what and all we have discussed here? Let me explain the same things in a table. Okay, let me explain the same things in a table. Guys, listen carefully. There are different channels for you, especially three channels I have discussed with you. A small typing mistake is there, you need to correct it. I have explained you about the DHPR channel, RYR channel, and Sarca. Three channels I have explained you. DHPR channel I have taught you, it is present on the T-tubule. The remaining two are present on the L-tubule. So here there is a small typing mistake you need to correct. So remaining two are present on L-tubule. Okay, the remaining are present on the L-tubule. L-tubule, RYR, and Sarca is present on the L-tubule. Now, later I will explain you, whenever you are having mutation of these channels, okay, whenever you are having problem with these channels, they will lead to certain diseases, guys. Here I have gave the diseases list. But one important disease is mutation of this RYR channel mutation will lead to a disease called as malignant hyperthermia. You will again study this in your pharmacology as well as anesthesia. So, RYR channel, if it is mutated, more and more calcium is continuously coming into the cytoplasm. That will lead to unnecessary muscle contraction. That condition is called as malignant hyperthermia. Because of this unnecessary muscle contraction, there is so much heat production in the body, that is called as a malignant hyperthermia. Okay. So DHPR, RYR, Sarca present on one T-tubule, remaining two on L-tubule. What is the function of Sarca? Have I taught you? Sarca, what it will do? It will sequester. See, it is sequestering the calcium back into sarcoendoplasmic reticulum. I have taught you. Okay, so with this I am ending the first part of the skeletal muscle physiology. The remaining things will be discussed in the subsequent video. In the next video I will be explaining about the different components. We have already discussed, but I will give you the more summary, the cross bridge cycling. Okay, important things which were already asked in the FMG exam, that I will explain you in the second video. Thank you.